So good morning. I want to welcome everybody here today, and it's my pleasure to introduce Phil Corlett today. We all know Phil, but I'm really excited to hear his talk and get an update on what he's been doing. Dr. Corlett was trained in experimental psychology, cognitive neuroscience, and psychiatry with Professors Trevor Robbins and Paul Fletcher at the University of Cambridge. And while there, he won a Wellcome Trust Prize studentship and completed his PhD on delusion formation in the brain mapping unit of the Department of Psychiatry. After a short postdoc, he was awarded the University of Cambridge Parke-Davis Exchange Fellowship in Biomedical Sciences, which brought him to our Department of Psychiatry to explore the maintenance of delusions with Jane Taylor and John Crystal. Here he was named a Rising Star and Future Opinion Leader by Pharmaceutical Marketing Magazine and an international mental health research organization, One Mind Rising Star. He joined the faculty in 2011, where he continues to explore the cognitive and biological mechanisms of delusional beliefs and hallucinations, as well as predictive learning, habit formation, and addiction. All of this is just incredibly interesting, and I'm looking forward to his talk. So please join me in welcoming Dr. Corlett.
Thanks, Stephanie, for the really nice introduction and the invitation. It's so great to share what we've been working on for quite some time now. Since I was in Cambridge, been interested in psychosis, which we sort of generally define as the profound departures from consensual reality that really trouble some people with psychotic illnesses, but nevertheless might be quite much more prevalent than we'd previously appreciated, and perhaps not distressing for some people. And so the work that we've been doing has really been trying to go back and forth between patient studies, studies in healthy controls, using brain imaging and computational methods to learn about psychotic symptoms, but also to use them to teach us something about how the brain works and how we all perceive and believe. Have no disclosures, sadly. But this work really builds upon a history of biological psychiatry, I think, that was much more bottom-up. This is a slide that I borrowed from Danny Weinberger, who's really the sort of doyen of the biology of schizophrenia. And Danny's plan, and I think the plan for the NIH for many, many years, was if we could only find the genes that confer risk for the symptoms of serious mental illness in large population studies, we could do a lot of basic biology on how those genes are instantiated in cellular and systems level processes, often in infrahuman species like rodents and primates. And somehow, magically, psychiatric phenomenology would just pop out at the other end. And I guess the work that we've been doing in the lab has been trying to take more of a top-down approach, to really talk carefully with people about the sorts of things that they're experiencing, to try and capture those things as best we can with interviews, but also with cognitive tasks, to really understand exactly how it is that brains give rise to, or are correlated with, or related to psychiatric symptoms, in the hope that we can back translate, right? We can use tasks that might be more amenable to being run in infrahuman species like rodents and primates, so that we can really make use of the wealth of new technologies and new biological information that we have available to really kind of ground and understand what it is about these genes that confer risk for serious mental illness. And the way that we've done it, and the way I'm going to talk about it today, is in terms of how causal beliefs and causal inferences and causal learning might relate to delusions, and how our perceptions of the world are driven by predictions and prediction errors, and ultimately how those things manifest as delusions and hallucinations. And when we think about computational psychiatry, I think many of us have a tendency to jump immediately to big data, really large data sets where we can use machine learning to try and figure out what it is that confers risk for serious mental illness, or how we might match patients better with treatments, making the most of huge data sets from all around the world. I guess what the work that we've been doing is a little bit more bespoke and a bit more focused on smaller data sets, and very much inspired by this little book by John von Neumann, who's really the father of game theory. This is a book published posthumously, actually. It's the transcript of a lecture at Yale that he never got to give because sadly he passed away. I've got my copy here. It's very, very thin. And in it, he wrestled with his understanding of computers. This predates personal computers, but it talks about the overall idea of computation and Turing machines and how they might relate to the types of computations that would be possible to conduct within a brain. And it's grounded in the Hodgkin and Huxley and Katz equations about how neurons might fire. And it's related very clearly to McCulloch and Pitts' work on how groups of neurons might connect together and form or conduct logical computations. The really interesting thing about the introduction to this book actually is in the beginning, he apologizes that he isn't a psychiatrist or a neurologist, but he's going to nevertheless wax lyrical about computation in the brain. I thought that was quite funny because it's gone on to be so influential for everything that we've done, certainly in my lab and more broadly in the field. So smaller data. So this is the first experiment that I ever had the pleasure of conducting with these three gentlemen, Trevor Robbins, Dave Theobald, and Jeff Daly. This was my undergrad research project in Cambridge. And it's what taught me that even really simple learning mechanisms like Pavlovian conditioning can go awry and make animals engage in really strange behaviors. So in this case, what we're studying is something called auto-shaping. What happens on each trial for this rat is he walks onto this little pad here to initiate the trial. This white box here is illuminated on this side of the screen, and a food pellet is delivered into this hopper here. So he learns that this white stimulus here is associated with food delivery, and he learns it over and over again, repeatedly. He actually learns it so strongly that he comes to substitute the food for this stimulus, and he'll start to approach and try to eat the stimulus on the screen. So excessively strong Pavlovian conditioning driving his behavior to the point that it's maladaptive. And indeed, if you withhold food, if he approaches, he'll still continue to do it. So strong learning, setting him awry in his thoughts and his actions. What we did was to drop cannulae into the nucleus accumbens, which is a region of the striatum, and manipulate dopaminergic and glutamatergic function. And we showed that if you knock dopamine out, you can stop the animal from learning, and likewise if you knock glutamate down. And that's not the case if you delay the infusions until after those memories, that learning had been consolidated. And at the same time, in Barry Everett's lecture series, I was learning that these chemicals, dopamine and glutamate, were implicated in the pathophysiology of schizophrenia. And it got me thinking, well, what if in schizophrenia, the symptoms, the sort of symptoms at the forefront of our diagnostic criteria, delusions and hallucinations, might be driven by too much dopamine, too little glutamate, driving people to learn and attend to and think about things that they really ought not to, and to consolidate that information too strongly and to use it excessively in guiding their everyday behaviors. And that's what really I've been following up on ever since. So I've used the word delusions a few times. This is our DSM-5 definition of what a delusion is. They're fixed false beliefs that aren't amenable to change in light of contradictory evidence. They've got lots of different themes. They're usually around one's relationship to other people in the world, on our sort of social status. They're typically deemed bizarre if they're implausible and not understandable by other people from your culture. And the distinction between delusions, as I mentioned, and other strongly held ideas is sometimes difficult to make. Now, in the definition, they talk about degree of conviction. What I'm going to show you today actually is the degree of distress with which delusions are held might really distinguish them from healthy but nevertheless unusual or odd beliefs. But the key thing for the work I'm going to present today is that we think about them as beliefs. So we can draw on the cognitive neuroscience of belief to try and think about how delusions might represent mutations of that belief process. This is an example. This is the Capgras delusion. It was first reported in the 1800s in Paris by two neurologists, but this is a 74-year-old married housewife diagnosed as psychosis not otherwise specified because she believes her husband's been replaced by an unrelated man. She refuses to sleep in the same room as the imposter, she locks herself in her bedroom, and she asks her son to procure a gun to protect her against this imposter. Now, I think it's really, really important that we remember that people with serious mental illnesses like this are 14 times more likely to be the victims of violence than the perpetrators, but nevertheless, delusions and hallucinations do sometimes drive action with tragic consequences, and I think that's something that hopefully this work might speak to in the future. It's certainly something we're really interested in following up on in the lab. Definition of hallucinations is much more simple. Percepts without an external stimulus in a conscious and awake state, having the qualities of real perception. There are a couple of problems with this. There are, of course, hypnagogic and hypnopompic hallucinations, which even some of you may have experienced. Those are hallucinations that you have as you're falling asleep or waking up. I certainly had them when I was writing up my PhD thesis and really stressed out. Here's an example. This is a typical example from the UK. A 21-year-old university student referred to University Health Services because he's hearing voices in his head. He tells his psychiatrist for about a year or so he hears people talking about him when he's on his own and there's no one else there. 2 male voices discussing him a couple of times a week, sometimes commenting on his actions. And interestingly, he builds a sort of delusional belief around these experiences. So he thinks that MI5, which is the security agency that James Bond is part of, are plotting to kill him. And I point this out because the data that I'm going to show you have some tensions and some difficulties, because the processes that lead to delusion formation might actually be different than the processes that lead to hallucinations. And we have some ideas about how to figure that out, and we've just got a new grant to try and work on that. But nevertheless, we don't have an answer readily available just yet. And we think we know a lot about psychosis because we can give drugs that block dopamine receptors in the striatum, and psychotic symptoms sometimes go away. Certainly, many of you, the clinicians of you in the crowd, will know that D2 blockers do pretty well against psychotic symptoms like hallucinations and delusions, except that in about 30 to 50% of patients, there's still some residual psychosis, despite them actually taking their drugs. And there may be a group of people who are what we call treatment-resistant or non-responders. These are data from Ollie House's lab at the Institute of Psychiatry in London, doing a PET scan to measure presynaptic dopamine release and showing that people who respond to the antipsychotic drugs that we have available actually have an excess of presynaptic dopamine ready to be released. People who are treatment resistant don't have that. And using a different type of imaging that measures glutamate in prefrontal cortex, this is called magnetic resonance spectroscopy, what Ollie found was that the people who are treatment non-responsive actually had an excess of glutamate in PFC. So even when people have similar phenomenology of symptoms, there may be different underlying neurobiologies, and there may be sort of different ways in which these symptoms manifest themselves that require different treatments. And ultimately, that's one of the aims of the lab, is to try and figure out who's in this population, can we identify them with tasks or brain imaging, and can we line them up with treatments more effectively. In terms of the neurobiology of hallucinations, we know quite a bit from functional imaging studies. This is a really beautiful study that Ralph Hoffman and Michelle Hampson reported a number of years ago now. Typically, what we do is ask people who are hearing voices to lay in the scanner and push a button every time they hear a voice. And that would be the data at this 0 point. But the interesting analysis that Ralph and Michelle did, because they're both incredibly creative people, is to try and track back in time and see what was going on in the brain prior to them reporting the hallucination onset. And so these different bands are different temporal profiles of activity evolving over time in different regions prior to the onset of hallucinations, during hallucinations, and afterwards. And I just point out a couple of things. There's lots going on cortically in terms of activations and deactivations, even in auditory cortex, prior to the onset, 3 seconds before people report hearing a voice. And those dopamine-rich regions where all of the drugs that we currently have available to treat these symptoms actually bind don't come online in terms of activation until 4.5 seconds after they've reported hearing the voice. And to me, that suggests that when we treat with these drugs, what we're treating is people's attributions about the things that they've just experienced, not the experiences themselves, nor the underlying biological mechanisms. With delusions, it's a little bit more difficult because they don't wax and wane on the same time scale as hallucinations do. These are just some data from a little meta-analysis that I conducted with an undergraduate student, mapping lesions, regions of damage to the brain that have given rise to the monothematic delusions like the Capgras delusion that I mentioned previously. So what I'm showing you here are the 2 hemispheres of the brain, frontal cortex, parietal cortex, temporal cortex, white matter, subcortical regions, and occipital cortex. And what I've done is place a line linking 2 regions together if they're damaged at the same time in a particular patient. And so the heavier the line, the thicker the line, the more likely it is that 2 regions are co-damaged. And what you can see is there isn't really much of a coherent picture that emerges except that there's something about the right hemisphere that seems to be particularly important. And that'll be something that will be borne out in our data too. What I think for both Ralph's work and for the delusion work is that we need a psychological theory to put some meat onto those neurobiological bones. And I wanted to go really, really basic. And so you're all familiar with Pavlovian conditioning. I think you've probably heard of it before. What Pavlov did in his experiments was pair a metronome. It wasn't actually a bell. It was a metronome ticking with the presentation of a food reward to a dog who was hungry. And over, every time the dog heard the metronome, he was delivered the food reward. And so after a while, he would start to salivate in anticipation of getting this food reward prior to getting it, just conditional on the presentation of the metronome. We think this may be a way of understanding how humans learn to associate causes and effects. I'll tell you about that shortly. And more recently, we've come to think that associations between perceptual stimuli in our environment, even when no reward is explicitly involved, might be associated through the same sorts of learning mechanisms. And the mechanism that I've focused on in my lab was really put forward by these two gentlemen, Bob Rescorla and Alan Wagner, who at the time in the 1970s were two psychologists, actually at Yale, over in the psychology department. Alan sadly passed away last week. And they developed a theory of how Pavlovian conditioning goes about that I've been able to apply to causal belief formation and more recently to perceptual learning. And this is their model. It's really an overly mathematical way of saying that we learn from our mistakes. So you would tend to associate two things in the world to the extent to which there's a mismatch between what you expect to happen and what you actually experience, colored by a couple of learning rate parameters that code the salience of the cause and the salience of the effect, if you like, how important those things are to you. And in a really nice review about this process by Anthony Dickinson and Wolfram Schultz, they say prediction error relates to the very essence of learning. No learning occurs when you're not surprised, when the outcome is perfectly predicted. And in a nutshell, our model of delusions is that these signals happen in the brain when they really ought not to. You have prediction error when you shouldn't, and that makes you attend to and learn about things that you really ought not to attend to and learn about, and you form your beliefs in order to explain away those experiences. So aberrant prediction errors drive delusions. You've probably seen this slide in almost every talk that you go to. Prediction error signals are manifest, at least in terms of learning about rewards and associating rewards with visual stimuli, in terms of the firing of midbrain dopamine cells deep in area A10. So when there's no association, you just get random reward, you see firing to the reward itself. However, after a while, cues that have come to be predictors of the reward show the firing. It's surprising because the animal doesn't know exactly when the cue is going to onset. And interestingly, they also show a dip below baseline in firing when an expected reward isn't delivered. We wanted to see whether or not we could extend this type of learning and look for these sorts of signals in humans with functional neuroimaging. And the way that we did it was to use a food allergy paradigm. So we get people to imagine that they're an allergist. Their task is to figure out which different foods cause allergic reactions in a fictitious patient. And so on each trial, we're going to show you a food that this person's eaten, and then we're going to show you whether or not they had an allergic reaction. Your task is to, when you see each food, make a guess and hold the button down for longer, the more confident you are that you think you're making the right choice. So if you're just guessing, you tap it a little bit. If you're really confident, you hold it down longer. So you see hamburger, and you see an allergic reaction. Every time you see a hamburger, you see the allergy, along with a whole host of other foods, but hamburger is the key food here. In the next phase of learning, what we do is show you hamburger paired together with a novel food that you haven't seen before in this task, bananas. You don't know about the allergenicity of bananas yet. And you see that these two things paired together lead to an allergy of exactly the same magnitude. And the question is, what do you learn about the bananas? Because everything is already fully predicted by the hamburger. There's nothing new to learn about the bananas. There's no prediction error, and therefore you shouldn't form much of an opinion about them. And indeed, that's what we find behaviorally. So this is work from Sarah Feinberg, who was a psychiatry resident and is now junior faculty member. Sarah tested a whole bunch of people on this task, actually in the psych emergency room. These were people who were controls, but nevertheless were in the psych ED for some reason other than having psychosis. And what you can see is they form an appropriate belief about the truly causal stimulus, and they don't have much of a belief about the bananas. However, if you're in the PsychED because you're psychotic, you don't learn enough about the appropriate cue, and you learn too much about the irrelevant stimulus dimension. And that's entirely consistent with our idea that there's prediction error signals happening when the hamburger and bananas are presented together that make you attend to the bananas and learn about them and give them causal significance. Now, we can do this in the fMRI machine, and we can track people's brain responses to having their expectancies confirmed or violated. And you'll see that a region of right DLPFC, pretty much the same one that was lesioned in all of those lesion cases, is engaged when you see something you weren't expecting and not particularly responsive when something that's predictable or run-of-the-mill happens. Unless you're somebody with first episode psychosis, in which case your right DLPFC, as well as your striatum and hippocampus and a number of other regions, show this interesting blurring of the distinction between surprising and unsurprising events. In people with first episode psychosis, different things seem important and important things seem different. And the blurring, the smaller the height difference between these two bars, the more severe people's delusional beliefs are. And this is work that I had the pleasure of conducting with Paul Fletcher, Gary Honey, and Graham Murray in Cambridge. So there's a correlation between how severe people's delusional beliefs are, taking into account all of their other symptoms, including their hallucinations, and the inappropriate engagement of this brain region in response to events that really ought not to be surprising. Now, one of the reasons I wanted to come to Yale was that John Crystal had been perfecting a technique to render people transiently and reversibly and safely psychotic for a short period of time. And that was to infuse them with a drug called ketamine. Prior to its miraculous antidepressant effects, ketamine was used as a model of psychosis and still is sometimes. These are the sorts of things that people report when you give them ketamine. So this is a healthy control given a ketamine infusion. Every time you left the room, I thought another person dressed in your clothes was coming back into the room. It wasn't scary, just another person doing your job, a little older in age and weighing a little bit more. When we do that in the scanner, on placebo, we show exactly the typical pattern of behavior in right DLPFC. It's engaged to things that should be surprising. It's not very interested in things that aren't very surprising. However, on ketamine, just like in first episode psychosis, there's an attenuation of the appropriate surprise response, and there's too much surprise to things that really ought not to be surprising. If you capture that by looking at the difference in the height between these two bars across subjects, just like in first episode patients, there's a correlation between delusion severity and that blurring of the distinction between surprising and unsurprising events, only in right DLPFC, not in any of the other regions of the brain, and only with regards to delusions, not any of the other symptoms. So we think some evidence that aberrant prediction errors are associated with and may even cause delusions to form. Now, as you know, we're pretty prone to unusual beliefs and conspiracy theories. I believe the last poll showed that 60% of Americans endorse at least one conspiracy theory. And as I mentioned, there's this idea that's coming out of computational psychiatry, but actually began here at Yale in the 1960s with John Strauss's work, that psychotic symptoms like delusions and hallucinations might be on a continuum from health into illness. We wanted to see whether or not those sorts of ideas were borne out in our data. These are data from Jim Van Os's study in the Netherlands, showing that even in the healthy population, about 3% of people, despite not needing clinical attention, would have symptoms that would be considered serious enough for a psychotic disorder. And more broadly, about 15% of people in this whole egg would have psychosis-like experiences. So we wanted to try and track those. And for delusions, we used something called the Peters Delusion Inventory. It was invented by Emmanuel Peters and Philippa Garrity in London. And essentially it has 20 now, there used to be 40, but they cut some of them out, 20 different delusion-like beliefs. Do you ever feel that your thoughts are alien in some way? If you answer yes to this question, it then triggers a series of further questions. Do you find that distressing? How convinced are you? And how much do you believe that it's true? And how preoccupied are you by it? How often do you think about it? What Emmanuel and Philippa found is that you can find people out in the world who are as convinced of these sorts of beliefs and are as preoccupied by them, but they simply don't find them to be as distressing as patients with a diagnosed psychotic illness do. And so what we did was capture the distress with which healthy controls held these unusual delusion-like ideas, and we regressed that distress back onto their right dorsolateral prefrontal cortical prediction error signal, exactly as we showed with ketamine, exactly as we showed with first episode psychosis. There's a relationship between delusion-related distress and inappropriate engagement of that brain region. So even in healthy people, there's something that looks like a psychosis-related brain signal that relates to unusual beliefs about the world. I think some evidence that the continuum idea might actually be true. Now, I promised you some computational psychiatry. It turns out that one of the great things about the Rescorla-Wagner model is you can model it in Excel. And that's what we did. So you can look at how beliefs form to the hamburger, the cue that was learned about first, and the banana. Right? No belief forms to the banana because there's no prediction error. However, if you add noise into the prediction error signal, you can make it learn inappropriately about the banana. And that's what we think is happening in psychosis. And this is just a little picture of what we did in the model to render the prediction error signal psychotic. And this is what happens in the brains of people during blocking trials if they have lots and lots of those odd beliefs and they're more distressed by them. So aberrant prediction errors, even during the blocking trials, that seem consistent with this idea that prediction errors are inappropriately engaged in people who have delusion-like beliefs, even in the healthy population. But like any story, there are, of course, wrinkles, right? So these are the data that I gathered with Sarah. These are some data I gathered with Genk Tech from people who are chronically ill, but nevertheless don't show inappropriate blocking. They still have delusional beliefs, but my contention was that they simply weren't as engaged with those delusions. They weren't at the forefront of their symptomatic picture. And my hunch, based on the distress work, was that people with chronic psychosis, although they endorse delusion-like ideas, they're simply not as distressed by them as healthy controls, nor as people who are acutely psychotic. And indeed, that's what we found. So when we run the Peters delusion inventory in people who are acutely ill, they find their delusional beliefs to be incredibly distressing. People with chronic psychosis still find their delusions distressing relative to healthy controls, but there's significantly less so. Now, that's probably because they're on medications, but again, being on medications and engaging with treatment might render your prediction error signal somewhat normalized, and it might be a marker for being currently or acutely psychotic. There's some data from basic animal studies. I'm just pointing out some from Beta Markadam now, but there's plenty of work, even here on the third floor at Yale, showing that if you distress an animal, you can acutely increase glutamate release in prefrontal cortex. And it's my contention that when people are acutely psychotic, this is precisely what happens. It's what we're doing when we give healthy people ketamine also. And that has clinical implications, right? So this is the mGluR23 positive allosteric modulator that failed in the replication trials. This is a reanalysis of those data, breaking people down into those who are within the first 3 years of their first psychotic break and those who are over 3 years since their first psychotic break. And what you can see is the mGluR23 compound that ought to decrease glutamate release, particularly in prefrontal cortex, actually helps and renders their symptoms improved. It doesn't help people who are more than 3 years ill, suggesting to me at least that the biology of psychotic illness might evolve over time and that the treatment approaches that we might take for people early in their illness might be very different than those later in their illness. And I think this has also implications for how we discover new drugs and the types of people that we might invite to be in our trials. I'm going to change gears now, though, and talk more about hallucinations and about the relationship between beliefs and perception. I'm really fond of this quote from Karl Jaspers, who's really the father of modern psychopathology, who argued that once you have a delusion, the trail is blazed and you're now prepared for significant experiences to occur everywhere that you have perceptions. The now dominant delusion motivates what you perceive in the future. I think that's true not only for patients with psychosis, but for all of us, that our beliefs about the world color what we perceive in a very strong way. And I'm going to just demonstrate that with this rotating hollow mask of Charlie Chaplin's face. It's going to rotate towards you in a clockwise fashion, but as you start to see the inside of the mask, something sort of strange and unusual is going to happen. This is a demonstration I borrowed from Richard Gregory. If I had the sound on, you could hear his British accent, which is much nicer than mine. Did you notice it? So it seemed as though the mask flipped directions rather than pointing inwards, it seemed as though it started to rotate in the opposite direction. I'll give it another chance to come back around. We think it seems like that to your brain because your brain has very, very strong prior expectancies based on everything that you've learned in your lifetime that faces point outwards. They very rarely point inwards. And light falls onto faces that point outwards in particular ways. And so your beliefs about faces and light override your percepts so that you see the world actually differently than it really is. In our lab, what we've been trying to show is that's what drives people to have hallucinations. Excessively strong perceptual beliefs that make them cut new percepts out of whole cloth. When there's nothing there to perceive, your beliefs are so strong that you perceive that there's something there. Now that's a visual example. I just wanted to give you an auditory one just to show that it works across senses. I hope this works. So what you're going to hear now is something kind of weird. Did you hear that? Sort of sounds like R2-D2. Okay. I'm going to play you what...
The camel was kept in a cage at the zoo.
And I'm going to play you that one more time. So the experience of the template renders this sine wave speech, which has been modulated to have certain frequencies change and the energy at certain parts of the signal altered, having a prior changes what you perceive. And now you can't unhear it. So again, we think this sort of learning mechanism, that prior that I just implanted into your brains, is what's driving people to have hallucinations. And indeed...
The camel was kept in a cage at the zoo.
I'm going to get stuck now. It was almost going so well. So when you test people who hear voices, this is a group of non-clinical voice hearers from a study by Ben Alderson-Day, Durham University in the UK. People with hallucinations in their everyday lives say that they can actually hear speech in those R2-D2 signals even before the reveal, even before they're given the template, much more strongly than and earlier than controls. And indeed, the earlier that they detect it, the more severe or the more common or frequent their voice hearing is in their everyday lives. So again, some hints that this sort of prior belief process might be related to hallucinations. And this idea isn't a new one. It dates back at least to Hermann von Helmholtz, although I think it's possible to go earlier. What Helmholtz argued is that what we perceive isn't merely just sensation that's incident on us. Actually, what we're doing when we're perceiving what's out in the world is drawing conclusions through practice and learning about what would need to be out there in the world in order for our sensations to make sense. It's not actually something that you do consciously or deliberatively. It's unconscious. And what Helmholtz suggested is that it's driven by the past association of ideas going on in the background unconsciously. And that fits with a set of ideas that myself and Paul Fletcher and people in my lab like Al Powers have been trying to develop over the past few years. The idea that we have brains in order to build a model of the world that makes what we're perceiving make sense, even in light of sort of degraded information. And the way that we might do that in terms of David Marr's different computational levels is that at the algorithmic level, we might do something like Bayesian inference, which is sort of like the Rescorla-Wagner learning rule. We have data or expectations about the world, hypotheses or expectations about the world. We have new data, and we reason about how to weight those two things based on how reliable they are. And that might be implemented in how our brains actually work. That is, we might send hypotheses about what we're perceiving top down. We might process data sort of bottom up and only send up the hierarchy things that conflict with our hypotheses so that they might update our beliefs. But the sort of tension between those two things is modulated by slower neuromodulators depending on which hierarchy we're in. That might be acetylcholine for the visual system. It might be serotonin, which is also involved in the visual system too. Or it might be dopamine in terms of our belief formation and reinforcement learning. Kind of fond of this idea that comes out of this way of thinking that everything we perceive is a form of sort of controlled hallucination. If there's some model of the world that's driving what we infer, the sort of balance between what's coming in and what's already in there is toward our models of the world. And I'm really fond of this quote from Pavlov that I was able to find from some of the translations of his lectures that said what the genius Helmholtz was referring to when he's talking about unconscious inference is the mechanism of the conditioned reflex. And that's something that Al Powers and I tried to set out and test. So Al was a resident at the time in the NRTP. He's now, I'm very happy to say, a faculty member, and it's just been such a pleasure to work on this with him. He's been absolutely brilliant. Al and I set out trying to study conditioned hallucinations, which was a phenomenon that actually Amy Arnston alerted me to when I first came to Yale. She said, you know, in the 1890s at Yale, they studied these learning mechanisms that gave rise to cause people to have hallucinations. 1895, I think, was the publication in the Proceedings of the Yale Psychological Laboratory by somebody called Seashaw. But Al and I focused on a more recent version from the 1940s by Doug Elson. And what Doug did was bring people to the lab and have an array of tungsten filament bulbs where he would put a very small voltage through the bulb so it would very faintly illuminate. And every time he did that, he'd play subjects a tone through a set of headphones. So bulb, tone, over and over again. And what happened was, after a while, after a good number of trials, he could illuminate the bulb, not play the tone, but people would report hearing the tone. So this is why hallucinations of the tone produced by sensory conditioning, pairing the light and the tone together. Now, this was the 1940s, so the sort of tools of modern psychophysics weren't really available to them at the time. The subjects in Doug's study might have been responding to demand characteristics, right? Doug's asking you repeatedly, did you hear the tone? Did you hear the tone? And you're wanting to be a good subject, and so, yeah, oh, yes, of course. So the great thing that Al did was to bring to bear really good psychophysics on this sort of task. So the first thing that we did, in the scanner now, with the scanner noise running, which, as I'm sure you're all familiar, it sort of sounds like a jackhammer going off, we're having people discriminate faint tones that are presented to them, and we're finding, using this psychophysiological quest procedure, the loudness of tone that we need to present so they get it 75% of the time, 50% of the time, 25% of the time, and of course sometimes we can play no tone at all. And then we're going to bring these different intensities, these 4 different intensities, through to a conditioning paradigm where we show a checkerboard that's paired with one of those tones repeatedly, and occasionally, and interestingly, we can show the checkerboard and not play any tone at all, and we can look to see how people respond. Do they report hearing it? And what's going on in their brain when they report hearing it. And like I said, we can take the tones of those different intensities to get around that demand characteristic problem. We can kind of scatter them randomly, or pseudo-randomly really, throughout the different training trials. So at the beginning, it's really obvious that there's a pairing between the checkerboard and the tone, but over time you can see there's more and more opportunity to make those conditioned hallucinations. And there's generally uncertainty about whether or not the tones are occurring. And I think that's really key. And we wanted to do this in 4 different groups of people. A group of people who had a psychotic diagnosis of schizophrenia or schizoaffective disorder who heard voices. A group of people with the same diagnoses who had never heard voices. A group of controls who had neither a diagnosis nor were voice hearers. And this interesting group of people who really get at that continuum idea. These are a group of people who hear voices every day, but do not meet diagnostic criteria for a psychotic illness. These are people who believed that they were psychic, or are psychic. They're what we call clairaudient psychics, so they're not clairvoyant, they don't have visions, they're clairaudient, they hear things, and they hear things at least once a day. And as scientists, we were somewhat skeptical of this claim, but we were open-minded. We thought, you know, there are people out in the world who have delusion-like beliefs that are as similar to delusions in the clinical case. Why not people who hear voices? And so what we did was bring people into the lab. This is my Manhattan plot of voice phenomenology. The key things to focus on, I'm sorry, it's a little bit blurred, but there are these different groupings of voice characteristics along the bottom here. So low-level characteristics, like the loudness of the voice, auditory qualities, the things that the voice says, the frequency with which the voice happens, and what you believe about the voice and how you engage with it. The only things that were different between the 2 voice-hearing groups in the study were mostly at this higher level. I think this one's actually misclassified. This is that you think your voices are deep, like thinking in words. I think I would put that up here rather than down here. But everything else, in terms of frequency, content, loudness, various other measures, were no different from patients with voice hearing in the psychic group, except that they believed that they could control the voices onset and offset at will much more frequently. They understood them in a more metaphysical or spiritual context, and they thought that they were generally a force for good in their lives, which some patients do, but that's not very frequent, or not as frequent in the patient group. And as I said, we didn't want to become parapsychologists. We were concerned that people were telling the truth about their voice hearing. And so we ran a series of tests actually from forensic psychiatry that we used to test whether or not someone's malingering around a criminal act. You say, oh, the voices made me do it. There are things that we can do. There are tests we can perform that give us some confidence that somebody isn't making up their voice hearing experiences. Both groups, both the patients and the psychics, passed all of those tests. So we're pretty confident that the voice hearing groups are actually hearing voices. So how do they perform on the task? If you're somebody who hears voices in your everyday life, you're about 5 times more susceptible to the conditioned hallucination effect than either people who have psychosis and don't hear voices and healthy controls who have neither psychosis nor voices. So you're much more likely to report tones that weren't present, conditional on the visual stimulus, if you're someone who hears voices in your everyday life. And that's particularly true at those lower stimulus intensities. So not at the 50%, not at the 75%. The groups start to diverge when there's more uncertainty about whether or not the stimuli were actually present. That's when you start to rely on your prior beliefs, and that's when those prior beliefs really kick in the voice hearing groups compared to the non-voice hearing groups. Your propensity to show the effect in the lab correlates with the severity of your voices in everyday life outside of the lab, measured here with the PANS, but we use lots of other different measures of voice hearing, and again, there's always this relationship. What we were able to do is, as I said, scan people's brains whilst we're doing this thresholding procedure, and as a sort of sanity check, the auditory cortex, particularly STS, its activity waxes and wanes with the intensity of the tones, which is nice. But what we can do is then use that region as a mask and look at what happens in terms of its activity when people are subsequently reporting tones that we didn't present them with. I think this is an objective measure that people are actually hearing the tones that we didn't present them with. That tone-sensitive region is engaged when they say, yes, indeed, I heard something, when nothing was presented, compared to when they correctly reject and say, no, actually, there was no tone. That's across all 4 groups, actually. When we look more broadly outside of that region, there's a whole swathe of regions that we think are involved in reinforcement learning, in belief formation and belief updating, particularly the anterior insular cortex, the striatum and the thalamus, but also the anterior cingulate too. And that was pleasing because, remember at the beginning I told you about all of those symptom capture studies where people are pushing a button to report when they're hearing voices. This is a meta-analysis of all of those studies showing what regions of the brain tend to be commonly engaged across the thousands of different subjects that have now done that paradigm. And when we overlay our data onto those data, it's pretty clear that we're engaging exactly the same circuit when we're doing our conditioned hallucination task, which is really reassuring, I think. Nevertheless, that doesn't meet what we were trying to do, or what I set up in the beginning, trying to understand exactly what it is that these regions are doing. And to do that, we turned to computational psychiatry. Al and Chris Mathis, who's just been a wonderful collaborator, created this model of how people might be responding to the task. The model has a perceptual side that makes sensory decisions, and that sensory information is read out into a decision-making aspect of the model that then says yes or no, stimulus was present or absent. And the model is hierarchical because it has different types of belief about the stimuli that might have been presented. So at this lower level, this is, do you believe that tones were occurring? This middle layer is, do you believe that tones were predicted by the checkerboards? And the top layer is, do you believe that those associations were changing over time? Which they were, right? We were making things noisier. If you remember that sort of pseudo-random curve, we were giving people more and more opportunity to break the association between the tone and the checkerboard. And these layers are linked together by different learning rate parameters. Particularly important is this one. This is a measure of how much people weight their prior beliefs, so everything that they've seen before in the task, on their current inferences. So that's the key measure of what we're trying to say here. If hallucinations are driven by prior beliefs, this thing should be higher in people who hallucinate. We wanted to make sure that we had the right model, so we actually used the model to simulate data, and you can see that it produces exactly the same effects, particularly the differences at the lower stimulus intensities, as in the real data. So we think we've got the right model. It's not overfit, and it's producing the things that it really ought to produce. In terms of people's decision-making parameters, there weren't any differences between the four groups. People who hear voices or people who had psychosis simply weren't just saying yes to everything. However, they were overweighting their prior experiences relative to people who don't hear voices. So both the psychics and the patients who hear voices tended to overweight their priors. They tended to have stronger beliefs that tones were occurring in the task compared to people who didn't hallucinate. They tended to have stronger beliefs that those tones were predicted by the checkerboards compared to people who didn't have hallucinations. However, the two patient groups now at the top layer of the model started to distinguish themselves. The psychics and the healthy controls were able to update their beliefs in light of the new evidence, in light of the changing contingencies across the task. The two patient groups, the people with and without voices now, were less sensitive to that manipulation. They didn't change their minds as much in light of the evolving evidence. So there's a nice double dissociation here where we can pick apart our four groups. We can say what's unique to voice hearing, and we can say what's unique to having a diagnosed psychotic illness. And that was present in the brain too. So if you tended to overweight your perceptual beliefs, you were somebody who heard voices, you'd have a much more robust response to conditioned hallucinations in the insular cortex. If you were somebody who struggled to update those beliefs in light of new evidence, you were much less likely to engage a robust response in the cerebellum and the hippocampus. So again, a double dissociation, not just in terms of behavior, but now in terms of neural circuitry underlying the phenomenon. Now, I'm really fond of this quote from the British statistician George Box. He says, all models are wrong, but some of them are useful. We wanted to try and see if this model was clinically useful. And we did that using transcranial magnetic stimulation, which is a procedure that follows sort of Faraday's law. If you have a very rapidly switching magnetic field near to a conductor, you can change or induce a current in that conductor. We're having a very rapidly switching magnetic field that's near to your scalp, and depending on the pattern of switching of the field, you can induce activity in regions or you can dampen down activity in different brain regions. What we thought we might do, in collaboration with the late Ralph Hoffman, was use transcranial magnetic stimulation, a 1 Hertz version, repetitive transcranial magnetic stimulation, to induce LTD over the auditory cortex. These are data from guinea pigs who are actually used in auditory experiments fairly frequently. I think that might be where the term comes from. If you give them a tone and then you give TMS, you can knock down the evoked response to that tone. We thought we might be able to do the same thing to decrease hyperactivity in the STS. We chose the STS first because it was the most accessible region. It's the one that's nearest to the surface. TMS doesn't reach too deep into the brain, although we now have a new coil that we think we might be able to get a little deeper. We might be able to modulate the insula and the cerebellum in the future, but right now we went for the STS. And we did that bilaterally in an open label study in a group of patients with schizophrenia. So what we did was bring them in. These are people who have chronic, intractable hallucinations, some of them hallucinating up to 80 times a day. We rated their symptoms. We rated their cognitive performance. We did that every single week for 4 weeks. We gave them a baseline fMRI scan, and we gave them daily TMS bilaterally without a placebo control, unfortunately, although we do have that capacity now, for 4 weeks, and then we scanned them again and rated their symptoms and cognition again at the very, very end. And this is what we found. Going through that procedure of having daily RTMS over auditory cortex significantly reduced the severity of their voices. I've now stayed in contact with these people for a number of years, and those improvements seem to be sustained. Their overall global symptom severity also reduces. So not just hallucinations, but also their delusions and negative symptoms too. And indeed, their cognitive performance, this was something we were worried about because we were knocking down activity in regions of the brain that were engaged by verbal cognitive processes. That actually improves. So their digit span, their ability to remember, say, a telephone number online when they don't have a pen to write it down, they could remember more digits than before we gave them the TMS. Their also verbal memory performance actually significantly improved as well. Interestingly, those two effects, the symptom effect and the cognitive effect, don't correlate with each other. So it suggests we're actually modulating independent processes. It's not just that you're hallucinating less so you can do these tasks better. It seems to be a more global improvement. In terms of what happens in the brain, we're looking at global connectivity now. looking at the coupling between every voxel in the brain with every other voxel. And pleasingly, what we found was exactly the same circuit that we engage with our conditioned hallucinations task that seems to be engaged when people hallucinate. Its global connectivity, it's the way in which it's talking to the rest of the brain actually significantly increases. And we wanted to focus in on one particular connection specifically, and that was the link between the STS and the anterior insular cortex, which is the part of the brain that was very much engaged very strongly by our conditioned hallucinations task. As you increase the coupling between Wernicke's area, STS, and the insula, people's hallucinations improve even more so. So the more that we change coupling within that circuit, the more significantly people's symptoms improve, which I think is quite pleasing and exciting. I'm probably going to stop now, not say too much more. What I've shown you is that early in illness, we think conceptual predictions about the world might fail, and that delusions might form as a result of the prediction errors that happen due to that failure. We think we can model some of that process with ketamine, particularly inducing excess glutamate release, perhaps in prefrontal cortex. Hallucinations alternatively seem to be associated with strong perceptual predictions, and we think that we might be able to treat them with TMS, inspired by this association between prediction circuitry and perception. I think in the future, one of the really exciting things that Al and I and Scott Woods are going to do is to try and use this task to predict the different trajectories that people take with their psychotic illness. It's very, very clear from the work with the psychics that it's possible to have sort of low-grade psychosis-like symptoms that are sustained throughout your life. But what about, is there anything that we can find out here that might distinguish between people who are going to take that path versus the people who are going to take that path? Indeed, is it the case that even really early on in the illness, in the prodromal phase, people who subsequently go on to have a chronic relapsing illness might actually have that poor belief updating process? That's something that we're hoping to be able to follow up on. But more acutely and more, I think, potentially practically, I've had the real pleasure of working with Al and with Claire Bien to think about ways in which this model might help us talk better to patients and people with lived experience of symptoms about what might be happening to them. As I've said throughout the talk, what you perceive, what you ultimately conclude, is a balance between what you expected and the data that you were presented with. And you sort of come up with an answer that's an amalgamation of these two things. We said that in situations when there's high sensory uncertainty, what you conclude is pulled towards your prior belief. And so perhaps when people are hearing voices, what we might want to do is help them find ways in which they can lower their sensory uncertainty. And with Claire, we learned from the Hearing Voices movement that there are lots of things that the Hearing Voices movement and Claire herself sort of advocate for that might help people reduce their sensory uncertainty about the world. They might want to avoid situations that are socially stressful, big crowds of people. They might want to engage in exercise or listening to music, which might structure their perception in a much more helpful way. And I think more broadly, this might be a way in which we can talk about the brain and talk about sort of state-of-the-art cognitive neuroscience with people who may have some reservations about neurobiological understanding of what might be happening to them. I think having this sort of psychological bridge might be a really great way of opening things up and having a better discussion about what might be going on, in which case we can learn from these people who are experts by experience and do better with the research that we do. And with that, I think I'll stop. I'll remind you again, with predictive coding, we think we can track people's current state and perhaps their progression. We can perhaps gain insights into novel treatments. We might be able to better match individuals to those treatments, which is what we call precision psychiatry. We might be able to tailor treatments to specific individual pathologies. And ultimately, I think it's important that we look transdiagnostically. There are lots of other people, other than people with a diagnosed schizophreniform illness, who hear voices, including people who have no illness at all. Thank you very much for your time. Thanks to the people who did the work and funded it. And just one more quick thing. Johnny Benjamin, who here he is getting his knighthood, is a mental health campaigner and advocate who I've been having a conversation with about voices and their impact on people. He's going to be coming here under a Poynter Fellowship, and we're going to continue that conversation here at Yale, and I'd love it if you'd join us for that. Thank you.